Good morning, and thank you to ARM, but more importantly, thank you to all of you who are attending today to hear our story. So Luminary Therapeutics kind of hails from a gene editing tool company where we have a lot of experience in both gene editing and cell therapy manufacturing. So as we look at kind of some of the problems that have already been mentioned in earlier discussions, we look at the high cost and limited patient access of effectively autologous cell therapy. We look at how CAR-Ts have really not had their day in solid tumors yet. And then we also look at blood tumors where we still have 30% relapse rates. So we at Luminary have been working on these challenges since we started the company. And I'd like to walk you through our data as well as our multi-indication approach with our CAR designs, and then some of our ongoing and future human trials. So this is a slide that everybody in this room, I'm sure, knows. It costs, $500,000 to treat a patient with an autologous CAR-T therapy. But more importantly, what really limits the access is a combination of just the logistics of engineering and creating autologous therapies. But something that's not talked about as much is the Medicare programs that most of these are patients of effectively don't know how to pay for these types of therapies. And so therefore, we really have limited access. So what's the answer to that? So Luminary's answer to that is an allogeneic platform, like all of us are trying to drive towards. And we do that in one of three ways. So the first thing that we use is our immune cell of choice is gamma delta cells. As being former people who've worked on engineering macrophages, NK cells, a variety of other types of iPSC cells, we feel that the gamma delta cells effectively are the best approach and haven't really seen their day in the sunshine yet, but we think they will. But more importantly, what we do is in our final product, there's two subsets of gamma delta cells, and the other gamma delta companies that are out there choose one or the others. We actually, in our final product, have both the V delta 1 and the V delta 2 subsets. And what you get with that combinatory power is you get effectively NK type killing, as well as you get more persistent and adaptive T cell motions in the V delta 1. So additionally, to make sure that once those cells are engineered and put into the patient, that your own immune system doesn't attack those, we've added an immune cloaking. And we do this through shRNA, some other things. And what we do is we tamp down both MHC1 and MHC2 restrictions just enough so that they are evaded by your natural NK cells and T cells. And then finally, we engineer those cells with a non-viral method that we created from our former company, which allows us a significant cost savings, but more importantly, allows us a significant time into the clinic. So with our first constructs that we took from an autologous standpoint, we were in the clinic within 19 months with a phase 1 IND approval because of using that non-viral gene engineering method. So does it work is always the big question, right? So NK cells have had challenges with persistence. iPSC cells have had challenges with effectively drift and that long, hard engineering process that Andy spoke to. Well, if you look at some of the other companies like Adicet and Immatics, they're showing some pretty good results. On the top graph, and I apologize that I didn't label this, but this is with CD19 cells. We can effectively engineer those cells with CARs, and we've put in about five or six different CARs. The top graph shows the CD19. We can get engineering efficiencies in the 50 to 60% range. More importantly, we keep our both polyclonal subsets of V delta 1 and V delta 2, but then equally importantly is we can expand those engineered cells up to about 2,000-fold expansion. On the bottom graph there in the green, you can see that we've just put our BAF CAR, which I'll talk about later, into these gamma delta cells and show that they're very functional. And we have a lot of data on serial killing that effectively shows that we can kill out to 10 or 12 different passages. But what's most important in the challenge that I put out to my colleagues and my fellows here is, we believe we can deliver 180 to 200 patient doses per manufacturing run and significantly reduce the cost of a patient product. And we're aiming in our organization to drop that down to $7,000 and be able to deliver patient products for $100,000 to really cut into that cost and make that a patient access product. So now coming on to solid tumors. We all know that solid tumors, at least so far, have shown little effectiveness with using CAR Ts. The whole reasons are behind that is some of the stuff that was just spoke to, tumor heterogeneity, having trouble here speaking, the ability to penetrate the tumor microenvironment, kind of the metabolic activity and the persistence of T cells. So our particular answer to this is a very novel dual targeting CAR where we effectively share the activation domain, but we split off the co-stimulatory domain. We've in-licensed this technology from a gentleman named Gianpietro Dotti at University of North Carolina. I would encourage all of you to look at this paper that I mentioned here in Nature Cancer, because it effectively shows some very, very impressive results in clearing solid tumors. So in the first graph there, we have mice that were been treated with this dual CAR using CSPG4 and mesothelium CAR and have tumor clearance on 3 out of the 4 mice all the way out to 80 days. When he combined GD2 and B7H3, was able to get clearance all the way out to 90 days in a neuroblastoma solid tumor, and more importantly, was able in the re-challenging mechanisms to demonstrate that the mice could not take the cancer back. We are currently working with a combination of CSPG4, and we have taken his technology and put it onto our gamma delta platform, and you can see that we get serial killing through round 4 that is very, very active compared to even the alpha beta co-stim CAR. So we're very excited about this particular product and believe that it will deliver great results in the clinic. So now turning over to blood tumors. We know that for a fact that several of our indications, primarily CD19 and non-Hodgkin lymphoma and multiple myeloma, you have relapse and refractory rates. You have probably a 30% relapse rate due to antigen loss in non-Hodgkin CAR T therapies. And on the BCMA, it's closer to 7 to 12% as opposed to 30%. But we also know that in the BCMA world, the 2 commercial manufacturers are having trouble making those particular products. So what is our answer to that? We have a single ligand CAR that is a BAF CAR that's connected by the BAF receptor, but it has 3 antigens on the particular CAR T cell. So therefore, it makes it much harder for the cancer cells to mutate away. We have a whole host of data demonstrating that we only need 1 of those antigens to be present in order to get cancer clearance. There's some additional benefits, and we don't have any foreign linkers in this ligand CAR, so that gives us a little bit better of a safety profile. We've demonstrated that this works in vivo models, effectively clearing both, and this I think is a multiple myeloma indication that I probably should have put up there. We have 2 phase 1 trials underway currently at Luminary, one for a basket study in non-Hodgkin lymphoma being run at Cleveland Clinic, and then we have a study for relapse refractory of multiple myeloma being run at Case Western. So we think we can make a significant impact on the current patient population that doesn't respond to CD19 therapies or single antigen BCMA therapies. So both of our CARs, our construct of our BAF CAR, because it has 3 antigens on it, has a whole host of particular applications, not only in oncology, but I think many people in this room have seen some of the papers on CD19's effectiveness against lupus. Our BAF CAR, we believe, has, because long-lived plasma cells do not express CD19, but they do express BCMA and TACI, if I remember correctly, we think we'll be able to clear a larger part of that B-cell compartment and bring relief to those lupus patients. Then over on our dual targeting CAR, we currently have a full exclusive license to this technology of splitting off the co-stimulatory domain and a full exclusive license to the CSPG4 CAR. We're working with CSPG4 and CD70, where we have a pathway on CD70, but we are looking for partnerships of companies that might have either single CAR constructs in B7H3, GD2, MUC1, or GFR. So I'd love to talk to any companies that are interested in kind of availing themselves to this particular technology. Here's our clinical pipeline for the next 12 months. So as I noted, we are moving everything onto our allogeneic platform other than our lupus trial, where effectively we're going to bring that to market and able to bring that to market or to the clinic in the next 4 to 5 months. And then everything down from there is allogeneic. And we will start with a head and neck cancer on the solid tumor side. And then on the BAF side, we will look to what our phase 1 trials are telling us to inform which indication that we might pursue. And then we hope to bring our BAF CAR for lupus onto our allo program. So we think we've made some significant advancements on these 3 challenges that are facing our industry. And we believe that we're going to be able to solve those challenges. We expect to have clinical data readout this year from our 2 phase 1 clinical trials that we're running and a head and neck cancer trial that's being run on autologous by Gian Petrodoti at University of North Carolina. And all of those things will inform and prioritize our allo efforts as we move towards filling in our IND applications in late 23 and early 24. This is our team. It was founded by myself and 3 scientific founders from the University of Minnesota. We were the founders of Beamogen Biotechnologies, which brought together this non-viral gene engineering tool. And here is our clinical team, Dr. Paolo Chemi. Our chief medical officer is Matt Spear. And then there are some of our regulatory people. Here's our intellectual property that we have at the company. And so then the other thing, we're out trying to raise money like everybody else, and I know this is probably not necessarily the right forum because there's not that many investors in the audience, but we're planning on trying to do at least 3 to 4 new phase 1 allogeneic clinical trials coming up in 24. So thank you for your time and happy to talk to all of you.